From the campus of Stanford University.
People are worried about data. They're worried about their privacy and their security. They should be. We need secure systems.
This is the future of everything.
But we can't have a system that closes that data off. It is too rich of a source of inspiration, innovation, and discovery for new things in medicine.
With your host, Russ Altman.
Today on The Future of Everything, the future of stem cell and gene therapy. Now, when we think about therapies for disease, we usually think about things like pills and IVs, right? And maybe surgeries. In fact, definitely surgeries. Pills they have the chemicals that can help the disease by normalizing your physiology or killing unwanted cells, either bacteria or viruses or cancer. Sometimes you have to do it IV because it won't tolerate your stomach and it has to get directly into your bloodstream. And of course, for surgeries, we're taking the tissue out, either the cancer or the infection or the diseased tissue. Just get it out of there as part of the therapy. But there are new options coming into view for therapies. And one such new option is gene therapy. If you inherited a gene that makes you more susceptible to a disease, or if, as they say, it runs in the family, can we fix it? There are some famous genes. There's the BRCA gene that's associated with breast and ovarian cancer. There's the Huntington disease gene that's associated with neurodegeneration. There are even genes for common diseases like type 2 diabetes, depression, schizophrenia, high blood pressure. Can we go in and fix them? Give you a new gene. You don't need a pill. You don't need an IV. You're just going to get a fixed version of that gene. That is now becoming possible. Another new therapy area is cell therapy and involving also stem cells. Many diseases result from bad behavior of our cells that either act wrong or die, and we'd like to replace them, either with ones that are perfectly good and work, or ones that are better than good and have had some defect repaired. So when you think about damaged tissue, you might think about type 1 diabetes, the so-called frequently child-onset diabetes, where the cells in the pancreas that secrete insulin die. Can we inject new cells that will take up residence in the pancreas and make the need for insulin and for all of those treatments for diabetes unnecessary? Can we kill cancers using our own immune system, either natural immune system, or can we kind of goose the immune system with special cells that are a little bit made out of us, but a little bit have special powers against cancer? So Professor Tony Oro, is the leader of a new center at Stanford University, the Center for Definitive and Curative Medicine. You gotta love that name. And he's a professor of dermatology at Stanford as well. He specializes in pediatric diseases, including skin cancers, and other diseases. Tony, why did you set up this center, especially with such a bold name, Definitive and Curative? And do you think that the gene and the cell therapies are now here and ready to go? Where are we on the promises of these technologies?
Yeah, thanks, Russ, and thanks for having me. I think when I started out my career, if you had if your gallbladder was bad, we can cut it out. If you have an infection, you give an antibiotic. But if you had a diseased tissue, if you're a child and you were born with inborn error of disease, we could actually figure out what it is, and then we pat you on the head and then send you on your way. And maybe
provide what you call palliative treatment.
Palliative treatment.
Like just make the symptoms a little better.
We can make the symptoms better, but we cannot cure the disease.
Right.
And so, but science has marched forward during my lifetime, and there's an amazing revolution which has emerged all around all of us. And so the center really is an organic collection of individuals here at Stanford and collaborators around the world that came to the realization that it was now time, in this time in history, that we could use cells as drugs and genes as drugs to actually develop a more long-term therapy.
Right.
And so rather than a palliative or just temporizing, rather we can actually, with one treatment, cure the disease. And so that's where the word definitive and curative. It's definitive meaning it's done for good. And in some ways, and then this is, it's March Madness air time, and so it is one and done.
One and done.
One and done. And so it's the one and done center, if you will, which is definitive and curative rather than taking, for example, blood pressure. You take blood pressure medicines starting at age whatever you are.
For the rest of your life.
For the rest of your life. Cholesterol medicines for the rest of your life. What if we could actually cure your ability to metabolize cholesterol, and essentially you could probably eat.
That's right. So in addition to helping the patient by just having them say you can now forget about this disease, it's gone.
Yes.
It also will have great economic benefits because the ongoing cost of disease could be severely curtailed in the best cases. And in fact, this has happened, as you know very well. We now have these treatments for hepatitis C. Hepatitis C used to be a chronic disease, and as of about 5-7 years ago, there are now definitive and curative ways to just abolish it from the system. I would guess that this was one of your inspirations.
Exactly. I think we as patients and as physicians, we've really settled for this ability to make the symptoms better, but not getting rid of the driving disease pathogenesis.
So what have been so there's two things we have to talk about.
Yeah.
What have been the breakthroughs that make you optimistic that now's the time to do this? And then the second question, which we'll get to, is what are the earliest diseases to go after? But let's wait on that. Tell me, what has been the scientific discoveries that make you bold enough to create such a center?
Right, and I admit it's bold, but I think it's bold in that we have to, we actually, because of these three breakthroughs, I'll tell you about, we have now, which are, first one is stem cell biology, which we'll get to in a second, but we understand now that the genome, which encodes all the genes in the body, 2% of those of the genome, it codes things that actually do things, and the 98% are actually instructions that make those genes go. And so understanding how that genome folds and unfolds with time to actually give different cells in the body helps us understand that there's the full panoply of cells, what they do, and how one cell connects to another. That understanding really is stem cell biology allows us to then take a cell that's an adult cell that's committed to a particular purpose, make it go back in time to form other cells.
That's the idea. So the idea of a stem cell is that it has not yet differentiated into a liver cell or a brain cell or a skin cell. It is what they call pluripotent or omnipotent. It has many, many possibilities. And if you can get those, then you can turn them into the cells that you need.
Exactly. And so the other thing is that they actually will self-renew. And so they're around for a long time.
Right.
And so if you inject some cells in your body, they go away right away. If a stem cell has a certain potential to then convert to other parts of the tissue and is around for a long time, that's the aha that's currently available that we can actually harness and use those cells for manufacturing purposes to make things that are useful, to engineer them in a way that for medical purposes. That's the first thing. The second thing is genetics. We now understand what the disease basis is because of the sequencing of the human genome.
Right.
And so now we can actually, in a sort of the precision health sort of way, we know precisely what the individual problems are, at least more and more, for individual diseases. So then, in fact, we now can go in and correct them in a way to make them better. And the last thing is tissue engineering, where we actually now can engineer the manufacturing of cells for clinical purposes at a level that you would want to put these cells into your body, my body, or your family member's body and feel confident about it. It's now reached a level because of the amazing engineering here at Stanford and other places that gives us confidence.
This is the future of everything. I'm Russ Altman. I'm speaking with Dr. Tony Oro about what we just heard, which are the confluence of three unbelievable capabilities that give us confidence that definitive and curative treatments are on the horizon. They are, just to review, make sure I was listening, our ability to identify stem cells and manipulate stem cells to get them into different states, new types of cells, the ability to understand the genetic basis for disease, and the ability to take all of this and turn it into a real engineering and manufacturing discipline. And I want to start off here because that's a big one. you put a small molecule like a drug, like Tylenol, aspirin in your
Right.
body. The FDA has taken a very close look at that company,
Yep.
how they make the Tylenol,
Yep.
how they produce it. Are they creating the molecule accurately, and are they making pills where the other components are also safe and not going to cause a problem?
Exactly.
Now that's a little tiny small molecule, but now you're talking about an entire cell.
You're talking about a living cell.
And we all know that cells can get into places where they're not supposed to be. They can grow into cancers if
They
they're
can
stimulated.
change.
So
Right.
talk to me about how that engineering discipline fuels the rest of the activities.
Right. And in fact, we just had our third annual Center for Definitive and Curative Medicine symposium yesterday, where we have it's a confluence of the FDA, the Food and Drug Administration,
Yes.
the regulatory board for these.
They need to be on board with all of this.
Industry, which is on board to commercialize, and then academics.
Right.
And so it's really a conversation, because the bar for using cells as drugs is exactly the same as for pills. They have to be safe, 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 safe. But when you take a pill, it goes in your body, and then you pee it out at a certain time.
Right.
Well, these cells live for a long time, and that's part of the definitive medicine. And so we need to understand and how that works. And then for the
Cells also adapt to their environment, so they may change after
Exactly.
they enter, and they may change in different ways in different patients.
Exactly.
I would guess.
And so we have to understand what are the boundaries for change. And that's part of what understanding how the genome works. How we need to understand what the barriers are to change. And so we want to put a cell that is stable, that's not going to do a Dr. Jekyll and Mr. Hyde on you when you put it in. When you say, Mrs. Jones, we've cured your skin disease, but now you have a leg coming out of it.
Right.
That no one really wants that.
Are people thinking about like suicide switches in these cells so that you can say, okay, something's going wrong, turn it off?
Right. So if we can engineer it correctly from our knowledge, then I feel like we don't need those. But there's a whole industry, especially where there are suicide switches or ways to turn, kill the cell. In case
So this is still a kind of a debated area within the field.
No, I think they're all there.
Okay.
So we'd love it to have actually where you have a perfect cell population, and we know enough about it. And that's why a lot of folks, including my lab, are really studying what are the possibilities. Sort of like if your child is 4 years old, and we want to know where they're going to go when they grow up. We have to then understand what are the environments they grow to, and then what are the possible jobs they can do. That's what we want to know with the cell. Okay. When you put it in a certain environment, what are the possibilities? And then, for example, one of the engineering things about genetics is using these CRISPR technology
Yes.
to actually correct the genes that are defective in those stem cells. CRISPR technology, as you know, is actually new and different, and so we want to make sure that it actually cuts and corrects only that one little spot, not the rest of the genome.
I definitely want to get into that. So let's ground this for people. What are some of the diseases that look like they are the best current targets for these kinds of technologies?
Right. So we focus right now on childhood diseases because that have no other possible treatment. In part because these diseases, if you correct them and you have save a child from a lifetime of a horrible disease, and then are accessible where you can see them. If you put a new treatment on, for example,
I
on the
know
skin.
that you're pointing to your skin.
Yes.
Yeah.
I'm pointing to my skin.
You're a dermatologist after all. Let's not forget.
And I love to see skin. And so if you have skin and you're trying to correct it, you can see if it works or not.
Right.
Or if you're putting these new treatments in the body, the blood is also another one that's actually very useful because you can measure the blood.
An observable tissue is a big plus at this point.
So it'd be a horrible disease with nothing else.
And that's also ethically required because you can't withhold other treatments that would work.
That's right. And one that's well defined, one where the target, where you can actually know a lot about the stem cells, and that you can see it and observe it with time. And so, for example, the disease we work on is called Epidermolysis bullosa, which is a horrible blistering disease where one of the 12 proteins that keeps the outside of your skin, called the epidermis, on your body. So when you hit a door as you're walking out, that friction, the reason why your skin stays on is because one of those 12 proteins is working. If, in the absence of those proteins, your skin will fly off. And at birth, it just basically, and these are wounds that never heal.
Okay, I want to hear more about that. This is The Future of Everything. I'm Russ Altman. I'm speaking with Dr. Tony Oro about cell-based therapies, and in particular, this disease we're just learning about, or I'm just learning about, Epidermolysis bullosa.
Right.
And so just let's go back, because what you just said, you said a lot there fast, and it sounded pretty bad. So when a patient comes to the clinic with this disease, what is their complaint? What are they noticing?
Well, often they're noticing is that they have wounds that don't heal.
Okay.
And so when you cut yourself, your body does a great job healing those wounds. So you have a cut for a little while, maybe an hour or two, and it hurts. Imagine that happen where you have wounds all over your body. They never heal. Imagine the pain, suffering, and then
Yes.
what happens is that then they often get chronic wounding,
Right.
genetic disease, and then will die of
And you
some
said
sort of
that,
cancer.
as you promised earlier in your comments, this is a disease for which we know a little bit about the genetic underpinnings.
Yes.
And you said there's a protein, one of the genes in the genome, that has a mistake in it, I
That's
guess.
right. And so if you can correct that mistake, the skin cells can now make the glue. And then when you then take those cells and then put them back on the skin, they stick.
Okay, so that sounds great, except I know that there are billions of cells in a fully formed, even in a fully formed baby, hundreds of billions.
Right.
So how are we going to fix? Do we have to fix all the skin cells? Do we have to fix just some of them?
Right.
How do you get over that barrier?
So that's a good question. The first thing we're going to try to do, though, is just to show that on a small scale level, that there's a little bit of correction.
Fair enough. So cure one cell before you cure all cells.
Cure one cell, make sure it works definitively,
Okay.
and then we talked about scaling manufacturing and engineering.
Gotcha.
Then you develop, and actually what we're doing here at Stanford in our Center for Definitive and Curative Medicine, we have an amazing state-of-the-art cell manufacturing facility to innovate the scaling and manufacturing of those cells to the scale you need for the patient.
All right. So tell me about, are there other diseases that are ripe for these kinds of technologies?
And so there's over 5,000 genetic diseases in the human body, more as every day as we sequence different patients, different conditions, based upon the things that we were talking about, for example, the blood, things like the boy in the bubble disease, where they have
immune deficiencies.
Immune deficiencies. And so actually the director of our center, Maria Grazia Roncarolo, was one of the first people in Milan to actually cure the boy in the bubble disease putting a virus into the hematopoietic stem
But this was a
cells
good virus that carried
that
a
carried
correction.
the corrected gene.
Yes.
And now using CRISPR, for example, we might be able to then go in and correct it that way. Sickle cell anemia is another one. Other types of immune deficiencies. And then it goes on and on and on. And so really, once you have the platform, the technology to be able to take the stem cells out, correct them, and engineer them, then you can then use that cell technology to use cells as drugs.
This is The Future of Everything. I'm Russ Altman. More with Dr. Tony Oro about cell-based and gene therapies, and particularly perhaps some of the ethical issues associated with these technologies, next on SiriusXM Insight 121.
What are you going to do? You're going to go to Google and you're going to type in
The Future of Everything with Russ Altman from the campus of Stanford University.
Welcome back to The Future of Everything. I'm Russ Altman. I'm speaking with Dr. Tony Oro about new therapies, especially stem cell, CRISPR-based therapies that have now become possible because of really a revolution in biotechnology. And Tony, I have to ask you about the ethics of all these things. So you mentioned CRISPR in our last segment, and you're using CRISPR to correct defects in genes so that you can do definitive and curative therapies. But we all know about the recent news from the end of 2018 where somebody went ahead and CRISPR'd a baby, tried to, I think with the general goal of giving them resistance to HIV, which sounds like a pretty good thing, but really it led mostly to a outrage because of an inappropriate process. You also mentioned that you're already engaging with industry and FDA. So tell me about how someone who presumably is trying to do things the right way, how do you interact and think about the ethical aspects of the technologies that are very powerful that you're dealing with?
Yeah. So I think technology in itself is neither good or bad. It's a tool that we can use for our society. What we need to do in any types of technology, any new technology, we need to then form groups in our society that then help to understand what an ethical basis of use in that society is. So, bottom line, no technology should be done as an individual, which was the first mistake that was made. It should be done in a group setting with large interdisciplinary groups from different angles of society to come together and decide as a community to understand how to use it.
So you're saying that one of the mistakes that this fellow made was not engaging in several communal conversations to make sure that what he was thinking matched what everybody else was thinking about whether it was ready for prime time or not.
Exactly. So one of the key parts of the Center for Definitive and Curative Medicine is that as we move these technologies from basically this could be a cure to this is a cure,
right,
we then need multiple conversations with multiple aspects of social interaction, including the Food and Drug Administration, the Center for Bioethics at Stanford, industry, and most importantly also our patient groups.
Right. Because
those are the folks that for all of what we're doing is targeted to help them.
So let me ask you, has the world, has the 1990s and 2000 world given you all of the groups and all of the processes that you need to do this work? Or are you seeing that, FDA is great, patient groups, of course, are critical. Are there missing elements where you're saying, I sure wish that I had a way to have this conversation, and I don't have it right now, because it's different from what we used to need when it was just pills and IVs?
Right. So I don't think society gives it to you. I think the scientists that actually create it, the technology, need to then form. And that, again, one of the reasons why we have the Center for Definitive and Curative Medicine is that we have some of the leaders of the folks that have created the technology. For example, Grazia Roncola was one of the first people to actually cure boy in the bubble syndrome in Milan.
Right.
Matt Porteous, also co-director, one of the pioneers of CRISPR technology, world expert and thought leader, and then more stem cell biologists come together and then really drive the public conversation on all levels.
So that's interesting because that's not necessarily what scientists have been trained to do. We're trained to be scientists, to make discoveries, report those discoveries, keep asking questions. So is it a welcome, well, two questions. Is it a welcome task, in your experience, for scientists to take on this very different role of kind of moderating a public discussion? And is it appropriate for them to be doing it? Are they getting the help they need from kind of professionals, the lawyers, the ethicists? It sounds complicated. No?
Actually, it's not really complicated because we have a tool. What scientists have developed are tools. And so what we need to do is figure out how to use those tools. And so you just ask basically your community. I think at Stanford, we have a long history of technology development and so I think the community here at Stanford is very used to understanding how to apply different technologies. Yes. Going back to the recombinant DNA, revolution and the Asilomar conference in the 70s. Those sorts of things where we need, and that's the kind of thing we need to have for these new technologies for stem cell biology, genome editing, to understand the ethical basis of it and what we need to, what we can and can't do as a society.
Yes, it sounds like there's even a role for historians to say, look, we've seen this before, or we've seen things like this before.
We've watched this movie before.
And here's what you do.
Right.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Dr. Tony Oro about emerging new therapies, the ethical and technical challenges. Let me just go to it. I like to end the show with hope, but this is a little bit in the opposite direction. What are the big worries? So for CRISPR, what are the big worries about the side effects of CRISPR therapies? And what are the big worries about the side effects of stem cell-based therapies?
Yeah. So the worries for any drug are relatively the same. Is it safe? And also when a company or a scientist says, I want to give to their patients, I want to give them this drug, the patients want to know how predictable are the outcomes in the body with time. And so the answer is exactly the same when I gave you Advil, when I give you an antibiotic.
Well, but wait, what I mean is, though, there's been a literature like for CRISPR potentially causing cancer. So no, just Advil in general is not associated with cancer risk. So I feel like...
But there's a long time history of knowing that it's safe.
Right.
And so we need to make sure and develop the rules and the tests so that and the technologies around this new technology
Right.
that can then help us predict when we give this drug to our patients, we know over time how it's going to respond. And it's the behavior of the drug with time in the person's body, in the site we put it in. That predictability is what we ask for, because then we know what's safe and what's not. We know that, and also the manufacturing of it, we know that it's pure.
Quality control.
So it's all quality control. And then so developing manufacturing technologies and quality control measures, and that's what the FDA is about. And that's what our Center for Definitive and Curative Medicine is, develop novel manufacturing and assays around this technology in a conjunction with an ethical framework to then know that we know it's safe. We know that when we give it to you, we know what we're doing, so that then we don't get to these horrible problems like people putting quote unquote stem cells in the eye to cure blindness.
Right, because there is a business right now.
There is a business about stem cells.
And is it in general not appropriate for these companies to exist? I mean, I know that it depends on the details, but what is going on with that industry?
Well, I think the FDA, the FDA and NIH convened a conference in December of 2017, in part because there was this burgeoning word stem cells and stem cell therapies that were not regulated, and where people were putting things they had no idea what they were.
Right, super scary.
No idea how they would respond, and then just shoot them in.
Neither definitive nor curative.
Exactly. or trust. So that non-definitive, non-curative, non-trustworthy.
So there's been progress at kind of shutting down some of the.
And that's what the FDA and the NIH are really involved in. They want to develop a way to then go back to the patients, the people we want to help, and say, now here's the framework we have for manufacturing that's pure and safe and predictable.
So one of the reasons that these companies did have customers, however, is the very understandable desperation of patients with terrible diseases. So can you give people some general advice? How do you know if you're dealing with a charlatan snake oil, and how do you know when you're engaging in something that really is pushing the boundaries of legitimate treatments? Because that's something everybody was going to need to know.
Everyone wants to know that. And so I would say the adage is true. If it sounds fantastical, it probably is.
Okay.
There's a long paper trail, both in the public domain. If you just Google it,
so if it's too good to be true,
it probably is.
Okay.
And so, but there's a long paper trail of publications and presentations that are around. The FDA, actually the NIH, the 21st Century Cures Act that the Congress passed has a hotline you can call and ask.
Oh, that's wonderful.
And so, there's a whole resource because the 21st Century Cures Act the NIH published, or passed, was in part, a small part, due to move these therapies into a safe realm. And so, call the NIH, call Stanford, call
your local university,
and
check
ask.
with your doctor.
Ask.
Because these resources are out there. And that's actually a very great way to end, because people are desperate with terrible diseases, and we don't want them to become victims as part of that desperation.
That's right. And so but the hope is that because of a group effort, that we can then develop these definitive and curative therapies for our patients where they're previously incurable diseases.
Thank you for listening to The Future of Everything. I'm Russ Altman. If you missed any of this episode, listen anytime on demand with the SiriusXM app.